You know, in science, there are some rules that you just feel are set in stone. They're the foundation for everything we think we know. But what happens when a discovery comes along that just completely shatters one of those rules? Well, that's exactly the story we're getting into today. So let's just start with a question that honestly sounds like a riddle. Can a protein that has no fixed shape, it's basically a floppy piece of molecular spaghetti, can it actually do a precise, complicated job inside a cell? It sounds like it shouldn't be possible, right? But that's the mystery we're about to solve.
Okay, so before we can break a rule, we've got to know what the rule is. For more than 100 years, biology has had this one central idea about how proteins work. It's called the structure-function paradigm, and the idea is really intuitive. A protein's specific job is determined by its unique, folded, three-dimensional shape. Think about it like a key and a lock. A key only works because it has a very specific shape that fits perfectly into one and only one lock. For proteins, it's the same deal. Their shape is the key. The rule has always been, no shape, no function. Period.
So our story starts deep inside the tiny, bustling city that is a living cell, where scientists stumbled upon a really strange puzzle involving some pretty critical infrastructure. See, every one of your cells has this amazing network of tiny highways called microtubules, and they're absolutely essential for, well, pretty much everything. They transport cargo around, and they're what pulled the chromosomes apart when a cell divides. They have to be built fast, and they have to be built right. And this brings us to our main character. This protein right here, called CKAP2. You can see it, it's that pink strand. CKAP2 is a master builder. It's known for helping these microtubule highways grow. But here's the crazy part. When scientists actually looked at its structure, about 60% of it is what they call intrinsically disordered. It has no stable shape. It's just floppy. So the question is, how on earth can this shapeless mess build such a precise structure?
So of course, the investigation began. If this protein has some parts that are structured and some parts that are just floppy, which part is actually doing the work? The researchers knew they had to find out.
And this image, this is the first huge clue. On the left, you see the entire CKP2 protein, doing its thing, binding to the microtubule network. Now look at the right. Here, the scientists took out everything except the floppy, disordered part. And look at that. It's doing the exact same thing. It's binding to the microtubules all by itself. So the really crucial point here is the shapeless part is the part that's doing the job. And this is where things get truly wild. This quote from the research paper really puts into perspective how radical this is. It says that for a protein to be an enzyme, a catalyst that makes a reaction happen faster, it was always thought that it needed a stable, folded shape. Finding an enzyme that's just a floppy chain, that was considered biologically impossible. Okay, so if the floppy part is somehow doing this job, how is it working? The main theory for other proteins that build microtubules is called the antenna model. The idea is that the protein has these long arms that reach out, grab the building blocks, which are called tubulin, and then deliver them to the end of the growing highway, kind of like a crane delivering bricks to a wall. So is CKAP2 doing that? Well, the researchers put that idea to the test. They used a really cool technique to see if the floppy part was actually grabbing onto those free-floating building blocks, and the result was a big, fat no. It just wasn't happening. Unlike other builders, CKAP2's floppy domain was not acting like an antenna. The mystery just got a whole lot deeper.
All right, so with the main theory totally debunked, the researchers moved on to the one test that could settle it once and for all. Was this shapeless protein a true enzyme, or was it something we'd never seen before?
Here's the key thing you need to know. A true enzyme is like a two-way street. It doesn't just speed up a reaction in one direction. It makes the whole reaction easier, which means it can also speed up the reverse reaction. So, if CKAP2 really is an enzyme that helps build microtubules, it should also be able to help dismantle them. And right here, this is the smoking gun. These images are called chymographs, and they basically track how long a microtubule is over time. On the left, you see those steep upward lines? That's the floppy domain making the microtubules grow like crazy. But then look at the right. When they took away the building blocks, you see these steep downward lines? The floppy protein is also speeding up the dismantling process. It's working in both directions. And we're not talking about a small effect here. It made the dismantling process happen twice as fast. This was clear, undeniable proof of enzymatic activity. So the conclusion was just inescapable. The verdict was in. This floppy, shapeless, disordered protein is a real, honest-to-goodness enzyme. That old rule, structure equals function, had just been smashed. So, what does this all mean? How does this supposedly impossible enzyme actually work, and why should we care? Let's take a look at how this one discovery is completely rewriting the biology rulebook.
This picture here shows the new model. So CKF2 isn't an antenna. Instead of acting like a crane grabbing bricks, it's more like a catalyst that hangs out right at the construction site. It gets to the end of the microtubule and it stabilizes that really awkward in-between moment when a new building block is trying to fit into place. By making that moment easier, it just lowers the energy barrier so blocks can pop on or pop off way more easily.
So why is this such a huge deal? Well, for one, it shows us a totally new way that cells build their internal skeleton. Two, it takes a biological dogma that's been around for a century and turns it on its head. And get this new method can be up to 50 times faster than traditional builders. And maybe most excitingly, it opens up a whole new frontier. Nearly a third of all human proteins have these floppy parts. How many of them are actually secret enzymes, just waiting to be discovered?
You know, a story like this is such a great reminder that in science, the rules we think are absolute are really just our best understanding for now. And it leaves you with this really exciting question. What other so-called impossible things are happening inside our cells right now, and what other fundamental rules are just waiting to be broken?